So hello, and thank you for the opportunity to give this talk today. I'd particularly like to thank my the organisers of the conference. So the title of my talk is Self-Healing and Self-Sensing of Concrete towards a Resilient Material for Life. And the general content is as shown here. I'll start off with describing why we need self-healing concrete. I'm going to give an overview of the Resilient Materials for Life projects from which a lot of this work has arisen. I'll talk about bacteria-based self-healing concrete, something about flow networks, self-sensing, and finally integration of self-healing and self-healing. The bacteria-based self-healing concrete is shown in bold, as that's the main topic of the presentation, and the majority of what you'll hear is based on that. Whilst I'm giving the talk today, I really don't want to proceed any further without making mention of the team at Bath that has been working on this area for the past 8 years through Materials for Life and Resilient Materials for Life. And some of the names on this slide will come up throughout the presentation. And certainly nothing that I'm presenting today could have been done without them. And essentially these 17 researchers are the co-authors of this presentation. Shown here are the academic staff, postdoctoral researchers and PhD students that have been involved with the research at the University of Bath. But even on top of this, there are many undergraduate and postgraduate taught students who have also contributed to this work on self-healing and self-sensing of concrete through their research dissertations. So to start off with, I thought it might be useful just to define the problem. Why do we need self-healing concrete? And obviously concrete is weak in tension, and due to a number of reasons, it may well crack during its service life. And this then permits the ingress of harmful compounds such as chlorides or sulfates that can cause longer term and serious deterioration of our concrete structures. And of course, you'll say, well, we know that, and that's why we maintain our structures. And we do invariably repair them from time to time. But I want to ask, do we really do that effectively? And I'd suggest not, because repair and maintenance of structures has problems. And indeed, current figures for the UK, where I'm based, show that at least half of the construction budget is spent on the repair and maintenance of mainly concrete infrastructure. And that's around about £50 billion every single year. But furthermore, these costs are compounded when the cost of disruptions, closing roads, closing railways, and other delays are considered. So the actual cost associated with repair and maintenance can actually be 10 times the actual figure of the repair itself. But actually, it's even worse than that, because the problem is many cracks start off small, so they're not spotted until they grow larger. So there's been many years in which harmful substances have been ingressing into the concrete before you've even noticed that there is a problem. And then, even once you've noticed that there is a problem, you can't just go out necessarily and repair the crack, because it may not have been budgeted for. So you've got to wait for the client or the asset owner to make that money available. And again, that means many more years, perhaps, when these substances are ingressing. So for this reason, I think, that it would be really useful if concrete could look after itself and repair its own cracks. And this is what I regard as autonomic self-healing concrete. And in the UK, work to develop autonomic self-healing concrete has been led by two large UKRI EPSRC funded projects. Materials for Life, that ran from 2013 to 2016, and its follow-on project, a 5-year programme grant called Resilient Materials for Life, that runs until the end of next year. RM4L is a partnership between Cardiff University and the Universities of Cambridge, Bradford and my own team at the University of Bath. And it's work through this collaboration that I'm going to be focusing on in this presentation, with of course specific focus on the work we've done at the University of Bath. So in terms of RM4L, just to get across some aspects of the size, it is a £5 million programme grant, and you can see the number of researchers that have been employed and have been working across the 4 universities. I've already shown the Bath team, but you can see actually the Cardiff and Cambridge teams are a little bit larger than that even. Importantly, we've had great support from a number of important partners throughout the construction industry, and they've contributed to the research that we're doing. And in particular, we've been supported by an external scientific board that comprises important researchers in self-healing concrete from throughout the UK, but also from TU Delft in the Netherlands, Ghent University in Belgium, and Shenzhen University in China. And we have an industrial advisory board that represents those that will ultimately be responsible for implementing self-healing concrete and self-sensing concrete in practice. And they come from throughout the supply chain and involve material suppliers, concrete producers, civil engineering contractors, consulting engineers, and importantly clients as well. And with this large team and the funding that we've obtained and with the support of our partners, we have a vision that by 2050 we can create a sustainable and resilient infrastructure containing smart materials and structures that self-regulate, adapt and repair without external intervention. And we intend that by the end of next year we will have a transformation in self-healing of construction materials through a biomimetic approach in which the material self-controls its own diagnosis and healing, and an evolution of next generation healing systems that cover diverse and complex damage and deterioration scenarios. To achieve that vision, the work in RM4L has been split into 4 research themes, which are shown here, and they address self-healing of cracks at multiple scales, self-healing of time-dependent and cyclic loading damage, self-diagnosis and immunisation against physical damage, and self-diagnosis and healing of chemical damage. And through RM4L, the teams at Cardiff and Cambridge, Bath and Bradford have been working on a number of different technologies to obtain this autonomic self-healing of concrete. And just a selection actually are shown on this slide. However, at Bath, which was of course where I'm based, we've largely concentrated on the use of encapsulated agents with a specific focus on encapsulating bacterial spores to obtain microbial-induced calcite precipitation within cracks, which I'll go into in much more detail in a few slides' time. Just waiting for the next slide to load up. Yep, there we go. It's got a video on it, so that's why it's taken some time. So that's the RM4L programme grant at a much higher level. We've also created this short video, or had the video created for us, which is aimed at the non-expert, and it explains what we're doing in a rather more fun way. So we'll just show this for now. Hopefully it will play.
One of the more polluting productions in the world is concrete structures. Over the years, tiny cracks form in the concrete, and to make sure the structures don't collapse, there is a need for even more polluting concrete production for maintenance. Researchers from Cardiff, Bath, Bradford, and Cambridge joined together in an adventure called Resilient Materials for Life, RM4L, and they came up with an interesting idea. Instead of adding more concrete into fixing the structure, what if the concrete would fix itself? RM4L is developing 4 main types of self-healing concrete methods. In the first method, microcapsules containing a healing agent are being developed to be mixed into the concrete. If a crack forms, it will rupture the capsules, releasing the glue-like agent that will glue together the microcracks from within. The glue agent is also used in the second method. Tunnels are built into the concrete with glue pumps attached to its side. If a crack forms, sensors signal the pumps to release the glue to fix the damage. The third solution is a shape memory polymer that's shaped into tubes and built into the concrete. If a crack in the concrete deforms the tubes, sensors placed inside the concrete will detect the damage and activate the polymers by heating them up, so the polymers contract to their original tube shape and pull the concrete together. The last healing method is by the inclusion of dormant bacteria and nutrients in the concrete mix. If cracks form and water penetrates the concrete, the nutrients are dissolved and the bacteria start feeding. This results in the precipitation of calcium carbonate by the bacteria and leads to the sealing of the cracks. The bacteria can also heal historical buildings and monuments. Self-healing concrete could be one of the solutions of tomorrow to reduce concrete-based pollution.
So, moving on from the video, as I said, at Bath, we've largely been exploring this latter aspect related to bacteria-based healing. So I thought it might be obviously useful to say a little bit more about how encapsulated bacterial spores work, how they can be optimized, and how they actually do provide self-healing, given it's the area we've been working at so intensively. So I'm going to explore some quick examples of work that we've been doing, which involves the fundamentals of bacterial growth, healing in colder conditions, some aspects of genetic engineering of bacteria, and a look at the availability of the growth media. So use of MICP for self-healing is fairly straightforward using bacteria. Essentially, we mix capsules into the concrete, and these capsules, often porous aggregates, for simplicity more than anything, they contain bacterial spores, and in some cases can also contain growth media. And the general concept is that the capsule is roughly the same strength as the concrete. So should the concrete crack, the capsule will also crack. And at that point, the bacteria spores will be released. And because of the bacteria that you choose to use, they're released into an environment that is conducive for them to germinate. And they will then induce the precipitation of calcium carbonate through a series of chemical and biochemical reactions. And I'll briefly explain these chemical and biochemical reactions, as the use of bacteria does create some fear in the eyes of some concrete technologists. But it's really actually quite a simple process at one level anyway. Essentially, bacteria are negatively charged on the surface, as I've tried to show here. This yellow blob is a bacterium. And they will attract free calcium ions in the concrete pore solution to their surface. And of course, there are many calcium ions in concrete. But of course, we can also increase the number by and therefore facilitate greater healing by adding further salts such as calcium nitrate to the mix, should we wish. In addition to this, the bacteria themselves are aerobic and will release CO2 into the concrete as a result of their metabolism. But we can also increase the amount of dissolved inorganic carbon in the concrete by adding organic salts, urea, or culture media. And actually the addition of urea is important here, as only some bacteria that we tend to call urealytic bacteria are able to break urea down. And therefore you end up with, you can talk about two separate pathways: the urealytic pathway that does use urea, and non-urealytic pathways. And I will be explaining or exploring this a little more in a few slides' time. But whatever the pathway, the presence of calcium and carbonate ions in the vicinity of the bacteria, which the bacteria will then acting as a nucleation point, leads to the formation of calcium carbonate, normally in the form of calcite, on the surface of the bacteria, as shown in this image here, which you've already seen, but actually last time I didn't annotate it last time. And this figure here on the screen shows then the typical healing of cracks using bacteria-induced calcite. The left-hand side is a normal mortar with a crack of 0.5 millimeters, formed at 28 days when obviously the concrete was hard. 30 days after the cracking, you can see that there's been some possible autogenous healing, but essentially the crack still remains. On the other hand, the mortar containing bacteria, which are called a bacteria-based self-healing mortar, here, you can see that the crack has healed after 30 days, and in fact it was largely sealed after just 7 days. So knowing how microbial-induced calcite precipitation works, one of the key things that we have been keen to do at Bath through our infrared has been to get a broader understanding of the capability of bacteria to precipitate calcium carbonate. So we've surveyed our local environment for the most effective bacteria. And we're very fortunate that Bath in the United Kingdom is close to many calcium-rich sites, such as of exposed limestone and caves in limestone areas. And within those areas, we targeted spore formers that could tolerate alkaline conditions, typical of concrete. And we also tried to distinguish our bacteria into ones that had that urealytic capability and those that didn't. And obviously this then allowed us to investigate which pathway was most relevant to our self-healing applications. And we investigated the kinetics of the precipitation of calcite and also the total amounts of precipitate that we could form. And we correlated this to bacterial growth and to changes in pH. And we saw with the urealytic strains a rapid alkalinization, which peaked at around about 2 days. And you can see that the theoretical maximum calcite, which is represented by the dotted line, was precipitated out actually by about 2 days, very, very quickly. Whereas the non-urealytic strains, they showed a more gradual increase in pH, and it took around about 9 days before we got to the amount of calcium carbonate precipitated was close to the theoretical amount that could be precipitated. But perhaps more important to that, we saw that the urealytic strains produced a much more finer and more homogeneous powder than the non-urealytic strains. And this is consistent with the idea that they were probably formed purely as a result of saturation kinetics changes, whereas the non-urealytic strains had large organic components embedded in the precipitate. In terms of self-healing of concrete, however, which is a bit more, is what we were genuinely interested in here, both the urealytic and the non-urealytic bacteria were capable of closing cracks in the concrete. But interestingly, we found that the non-urealytic bacteria, at least in the conditions that we tested them in, worked slightly better, which was to some extent a surprise. But it has meant that this is the pathway down which most of our research has proceeded. And also I think we found that the difference in the morphology was actually part of the reason for that. And we think that the gooey organic type precipitate that you get with the non-uretic bacteria is actually slightly better at sealing cracks. Now, you might be asking, well, why is finding new bacteria and understanding the mechanisms all that important? Well, I just want to take a step back now to 2015, so 6 years ago, when me and my team were relatively new to the field of bacteria-based self-healing concrete. And as part of the earlier Materials for Life project, the M4L project, we were required to undertake a site trial to demonstrate that bacteria-based self-healing concrete would work in practice. The site trial took place in South Wales, which is in the west of the United Kingdom, and it was carried out with the help of Paine, a major contractor, a major contracting engineer, and it was done as part of a major upgrade to an important link road, the A465 in Wales. We did the work in conjunction with our colleagues at Cardiff and Cambridge universities, who were testing some of the other technologies that I've already mentioned, and altogether 5 walls were made, and one of those was used for our bacteria-based self-healing concrete technology. And this wall here is 1 metre wide, 1.8 metres high, and 150 millimetres thick. Following a number of lab trials, we developed a concrete with the required design strength, appropriate consistence, and one that adequately provided self-healing in laboratory conditions. And the mixed proportions that we used are shown here. And the self-healing component, of course, is coated perlite containing growth media or nutrients, and coated perlite containing spores. The growth media was a mixture of calcium acetate and yeast extract, whilst the spores that we used were Bacillus pseudofermus, or at least they were known as Bacillus pseudofermus at the time. I've noticed that they've now been recently renamed alkali halobacillus pseudofermus. And the coating, so yeah, these were embedded within perlite, and then the perlite itself was coated with a double layer consisting of sodium silicate and cement paste. I think the only other thing really to notice about this, the mixed proportions, is that the cement we use is a CEM2BV, which means that it was a Portland fly ash cement. Due to issues with actually making a large number of spores, we didn't necessarily have the scale-up capability at the time. We decided only to add the bacteria-based self-healing concrete in the area of the wall where we knew the crack would form. So this was a section of 500 millimeters in height, 1 meter in width, and again 150 millimeters thick. But we knew that the crack would form somewhere just there. And that layer of bacteria-based self-healing concrete was mixed on site by me and a colleague. There I am. But all the other work was done by contractors, including the placing and the compaction of the bacteria-based self-healing concrete. And also, obviously, the concrete, the standard concrete that sat below the bacteria-based self-healing concrete and above it was supplied by a local ready-mix concrete supplier. And the truck you can see in the background there. And I think this is important because, in general, what we were trying to avoid was this being simply another university lab trial where university researchers have done this thing and it looks really good. What we wanted to demonstrate was that this was something that could be done on site. There was no reason why self-healing concrete technologies were not already available and could be done by contractors. And so therefore the vast majority of the work, other than, as I said, the actual mixing of the bacteria self-healing concrete, was done by contractors from Costain and by a ready-mix concrete supplier. And this is the end result. This is our bacteria-based self-healing concrete wall. I'm shown here with the postdoctoral researcher who assisted on the work. That's Dr. Trupti Sharma, who is a biologist by training. So she was a little bit out of her comfort zone on a construction site, but she did a fantastic job. And in this image you can clearly see where the bacteria-based self-healing concrete is, because that concrete is slightly darker than the rest of the concrete, and that's because it was made with Portland fly ash cement, whereas the rest of the wall was made with Portland cement. And this slide shows the finish that we achieved. And in my view, and obviously I'm biased, but in my view the finish of the bacteria-based self-healing concrete here is better than that of the normal standard concrete provided by the ready-mix concrete producer. And that's either because I'm brilliant at making concrete, or it's because the self-healing agents improve the consistency. Probably the latter. But this is important because when you tell engineers and clients that you want to add bacteria to their concrete, one thing they often worry about is that, what's it going to look like? What are the aesthetics? And clearly, the aesthetics of self-healing concrete are not a problem. It looks very nice. It looks as good, if not better, than normal concrete. So, 28 days after we cast that wall, and the other walls at the same time, they were all deliberately cracked. And this is different from many case studies that you may read on self-healing concrete, where they make self-healing concrete but they don't ever actually crack the concrete, and therefore you don't actually ever see whether they will self-heal or not. In our case, the walls were cracked. They were then left to heal, whilst of course being monitored for 6 months as well. And since the wall was cast, actually, I haven't told you this, but the wall was cast in October. So it was obviously, it was then left for 6 months. So it was left to being exposed in a South Wales winter. And South Wales winters are wet and they're cold. And actually this image shows the walls on a fairly nice day, but you can still see that there it is. Hopefully there is a crack somewhere there, and we were hoping that it would heal. And after 6 months, we came back to see all the cracks had healed, and oh dear, that doesn't look great, does it? The cracks are clearly still visible and don't appear to really have been closed at all. So what had gone wrong? Well, our chief suspect was temperature. Indeed, the average temperature throughout the site trial was around 7.5 degrees centigrade. And clearly a potential issue here was that we had used the mesophile Bacillus pseudothermus. As many groups around the world do, they use mesophiles. And let's not forget, our use of that bacteria in the lab had been absolutely fine. All concretes we'd made in the lab had self-healed. However, the problem with mesophiles is that in general they're not active below 15 degrees centigrade. Therefore, clearly in hindsight, and hindsight's a wonderful thing, it was never going to work in a wall in a South Wales winter. Clearly what we should have used was a bacteria that would work at a lower temperature, such as a psychotroph or even a psychophile. So, that's a bit of history. Let's now come back to the current research that we're doing as part of RM4L, where we have taken a look at this aspect of the performance. And this slide here shows some bacteria-based self-healing of cracks in a mortar at 20 degrees C, symbolised by the red thermometer, and at 7.5 degrees, symbolised by the blue thermometer. And Bacillus coni, which we've used here, is again a mesophile bacteria. And what you can see is that the healing in the warmer conditions at 20 degrees C has been absolutely fine. The Bacillus coni have healed that crack perfectly. However, in the colder 7.5 degree temperature, the healing is significantly, well, it's non-existent. There's maybe a little bit of something there, but essentially the crack is still there. It's not worked, which is exactly the same as the situation we found with the site trial. But consequently, one of the things that we've then done with that library of isolates that I talked about before is that we've identified at least one isolate that's capable of sporulation and precipitation in minimal media at low temperatures. And one of these, which we've called Psi-39, has demonstrated effective healing at low temperatures. And this, of course, is incredibly promising in terms of practical use of self-healing concrete, particularly in the UK, where we do have this temperate climate. And of course, indeed, the fact that we've found bacteria that work at different temperatures means that we can consider making mixed communities of bacteria that permit healing over a range of temperatures. However, when we wrote the RM4L proposal back in 2016, we did have some concern that we wouldn't be able to do that. We wouldn't find bacteria suitable for all conditions in which healing might be needed. So we considered whether it would be possible to take, for example, a bacteria that thrives at low temperature but has no calcite precipitating capability, and then give it that capability. So our microbiology team took a model organism, Bacillus subtilis W168, which has low intrinsic UAE's activity. It has poor calcite precipitation ability. And we used that as a chassis and then added to it various genes from a better calcite precipitating organism. In this case, Bacillus paralicheniformis. And of course, this is the wrong type of conference to look at this in detail. But we can be clear that when we look at the genome sequence of Bacillus subtilis compared to Bacillus paralicheniformis, it has certain genes missing either side of the UAE's structural genes. And essentially what we did was slowly over time add these genes back into Bacillus add these genes to Bacillus subtilis. Actually what you do is you take out their UAE structural genes and gradually put in that aspect, that part, then that part, then that part, and then you can do one with everything. And what we showed was that by adding these genes, you can get an increase in urolithic activity, which is important in terms of the urolithic form of calcite precipitation. For example, here, the red line is the behaviour of the Bacillus paralicheniformis, whilst the green line is the Bacillus subtilis. So you can see the Bacillus subtilis has very little UAE's activity. Bacillus paralicheniformis has a reasonable amount of UAE's activity. But then as you start to add these genes to the Bacillus subtilis, you start to form these brown, black and blue genetically modified bacteria that actually work better than both of the environmental bacteria. So it shows that it can be achieved. We also looked at the calcite precipitation ability of these genetically modified bacteria. And it was actually really interesting to see that the morphology of the calcite produced is dependent on the UAE's genes that the bacteria had. And in fact, with you can get even these very interesting shapes of calcite, such as these flattened calcite sheaths and this dumbbell morphology as well. So that's the bacteria sorted. We've got bacteria that can work at different temperatures, and if not, we can give bacteria that capability by genetically modifying them. What about the growth media? And because of the way self-healing concrete works, we do need to ensure the availability of calcium ions to precipitate the healing of the concrete. And one important question is, can we add the growth media directly, or does it need to be encapsulated, like I did in the site trial, for example? And this is an important question because the encapsulation of the growth media, which is a calcium salt and yeast extract, for example, is problematic because it takes up space that could be used by much stronger aggregates. It also increases the cost of production, and it potentially limits the amount of healing that can be achieved because you can't get that much calcium into the concrete. Because of this, you'll find that many research teams throughout the world doing self-healing concrete do not bother to encapsulate the growth media. And indeed, we at Bath prefer not to do so if we can avoid it. But perhaps we just need to, again, just look at exactly what's going on. We know why, how does the growth media work? An essential element of the growth media, as I say, is the calcium salt. And when you add that directly to the cement matrix, it dissolves. And it forms an excess of calcium hydroxide. And it's the calcium hydroxide that forms the long-term supply and reservoir of calcium in the concrete for healing to take place, should cracking occur. But of course we know from basic concrete technology that should the concrete carbonate during its service life, then the calcium hydroxide will be consumed and turned into calcium carbonate. At that point, of course, calcium carbonate is not in a position where it provides a reservoir of calcium ions. So the question is then, do we still get healing if the concrete has carbonated? So we've looked at this as part of RM4L, and this slide shows the normal lab situation where the concrete has not carbonated. And here we can see the relationship between the healing of the concrete and time. And we can concentrate on two self-healing concretes. One where the growth media has been added directly, which is the blue line here. And one where the growth media has been encapsulated, which is the green line there. And you can see that we get healing in both conditions. So it doesn't really matter. In non-carbonated concrete, generally speaking, as used by most people in the lab, it doesn't really matter whether you encapsulate the growth media or not. You could argue that based on these results here, that the direct addition, the blue line, is a little bit quicker. But the situation when concrete is carbonated is much different. And this, of course, is the situation the concrete is carbonated before it's cracked. And here there is, you can see, again, the same colour system. So the blue is where the growth media has been added directly to the concrete. And you can see there's very little healing has taken place. So now the only effective way to get healing is, again, the green line, where the growth media has been encapsulated before being added to the concrete. So essentially what we've suggested is that you end up with two potential systems. You've got a simple technology consisting of encapsulated bacteria, and that would be used for concretes that don't carbonate before cracking occurs. And then you've got a more complicated system where you need to encapsulate the bacteria and encapsulate the growth media, and that would need to be used where the concrete is likely to carbonate in what we would call an XC environment in Europe. But the fact that the growth media to facilitate self-healing may not always be available is, of course, a challenge. But we've kind of come up with a solution to that just now. But there are actually further challenges with an encapsulated bacteria-based system. We need to acknowledge that the the capsules can be quite large, and so they won't necessarily distribute homogeneously throughout the concrete, and consequently there is a real risk actually that a crack may miss them entirely. People have been looking at using microcapsules as a means of solving that problem. In fact, we've done that at Bath as well, but that again increases the cost of making self-healing concrete. There's a further problem that if you've got a encapsulated system, then the quantity of bacteria and growth media is fixed, and therefore it's liable to run out. Now that may not be a problem if you've got a single crack forms, that crack heals, end of story. But what if the crack occurs and comes back? What if you've got a situation where there can be repeated cracking? You're not going to be able to get repeated healing potentially in such a scenario. And there is actually a fourth problem which I've not put on here, but right at the beginning of this presentation I mentioned that the basics of our encapsulated self-healing works is that the capsule should have the same strength as the concrete. But that means we need therefore different types of capsule for different types of concrete strength, which is not necessarily all that easy to achieve, especially given that the strength of the concrete is not fixed. It changes itself with time as the concrete hardens. So, at that point we can say, well, hang on, let's take a step back. What are we actually trying to achieve here? And indeed at the start of M4L and RM4L, we were inspired to develop self-healing concrete based on a biomimetic approach. But of course the human body, and that of other higher animals, does not contain finite amounts of healing material contained within capsules. We have a vascular network that runs throughout the body, that's constantly topped up, and that can deliver healing wherever we should happen to cut ourselves. And there's no reason really why we couldn't use such a system in concrete. And this picture shows how such a flow network could be installed in concrete using shrinkable polymers. And this is something that my colleagues at Cardiff have been researching quite significantly. And indeed, as part of the M4L site trial that I described earlier on, Cardiff actually did put into our bacteria-based self-healing concrete wall a vascular network that would have allowed us to put further healing materials, which could again be bacteria and growth media. Additional bacteria and growth media could have been pumped or allowed to flow to the site of the crack and instigate either the healing or actually to top up the healing that we've already achieved. And I must say that, personally speaking, I think that a vascular flow network is a brilliant approach to the problem of self-healing, because it's so much more biomimetic, it's so much more closer to the way humans and animals solve the problem. But one of the challenges, and a major challenge with using vascular flow networks, is that they may require some human intervention. For example, we can only pump our healing agent through a vascular flow network if we know that the concrete has cracked. And this brings us, of course, back to our earlier maintenance problem and the fact that I was saying we don't often spot cracks in concrete because they're small, and we don't spot them until they're much larger, and therefore at that point releasing the vascular flow net, it might be too late. What we need is some way of being able to release the healing agent through the vascular flow network at the moment the crack first occurs. So something we've been working on at the University of Bath, again through RM4L, is to try and integrate the flow network with a self-sensing system, such that the concrete intelligently recognises that it has cracked, and so that it responds itself by diagnosing the crack and then effectively activating the healing system. And I think this brings us much closer to having an intelligent concrete that can sense, reason, and respond to its environment. And of course, this was the ambition that we've set out for RM4L for infrastructure in 2050. So to obtain concretes that recognise that they've cracked, or self-sensing, self-diagnosing concretes, as we tend to call them, we've used two methods, both based on the electrical properties of concrete. We have external sensing using piezo sensors, and intrinsic sensing using carbon-based powders and fibres. And actually, initially, and what I'm going to describe right now, is the work using PZT sensors, because that has so far proved the most successful in identifying cracks, and also actually in identifying healing. So this slide shows sort of typical piezo sensors and how we attach these to the concrete, at least in terms of doing it in lab conditions. We still need to prove this in real conditions yet. And we can crack the concrete in bending, and if we wish, we can even simulate healing by using compression. And as we load the specimens in bending, we can measure the impedance response over a range of frequencies. And what we see is that the impedance response changes from that in a pristine state, which is shown in black, to through loading through to a finally cracked stage, which is shown in red. Now, you're possibly looking at these images right now and say, well, I can't see any difference. Yeah, okay, because the changes in the impedance are actually quite subtle. But with some quite innovative analysis and computation, you can read a lot into these responses. For example, if you plot the impedance response at a given load against the response when it was in the pristine stage, you get the graph shown at the top here, where there was a fairly good linear relationship. And if you determine the coefficient of correlation, obviously it would be 1 for the pristine stage. And as you start to load it, the relationship between the impedance response and the pristine stage deviates a little bit, and by the time it's cracked, it's a little bit away from 1. But again, you couldn't really use that all that significantly as saying, well, the concrete is definitely cracked. But if you then actually plot the first derivative of the impedance, and then plot that against the pristine stage, you get the next graph shown here, where again for the pristine stage you get a R-squared value of 1, but as the concrete cracks, the R-squared value deviates away from 1, but so as it's loading it deviates away from 1. When it's cracked, there is no relationship between the impedance response and that of the initial, or very little correlation between the crack stage and the initial pristine stage. And I think at that point you could probably very clearly say the concrete must have cracked. And now maybe this is just a step too far, but there is a way to integrate the technologies that I have just described in order to get an effective self-healing concrete which has both encapsulated bacterial spores, a vascular flow network and sensor technology. So step 1, you could start off with the basic bacteria based self-healing technology, which can be used to autonomically self-heal any cracks in the concrete. But as it's doing that, it can be monitored by the sensing technology. And if it's considered that the healing is not sufficient, because, for example, differences in the impedance response, through differences in the impedance response, as we've just discussed, then the embedded flow network could come into play. And the sensors could trigger the remote the remote healing agents such as oxygen, growth media, further bacteria to be pumped to the site of the crack to assist the bacteria-based self-healing concrete. And then it goes back through the sequence again until a point at which the sensing technology says, the self-sensing system says, the healing has now occurred and everything can stop. As I say, that's one way to do it, and there may be simpler solutions. So at that point, I'm going to conclude and state that some of the work that we've done, we've demonstrated that the use of bacteria is a reliable way of recovering the performance of cementitious composites after cracking. However, it's necessary to consider specific environment and exposure conditions when selecting the correct bacteria and the growth media. And bacteria-based healing can be complemented with vascular flow networks and self-sensing capabilities in order to maximise the healing capability. So that's just a tour of some of the work we've been doing at Bath on autonomic self-healing of concrete as part of our RM4L research. As always, thanks to our colleagues at Cardiff, Cambridge and Bradford universities for their contributions as well. So thank you very much, and I wish you all a good day. Thank you.